As soon as I find your name back on our list, which was here, there we are, I'm going to make you a co-host. And Dave, would you like to introduce our presenter for this evening?
Sounds good. All right. We have Erin Posthumous. Am I saying that correctly, Erin?
Posthumous. It's fine. Close enough.
Posthumous. All right. Well, she manages the USA NPN projects that inform decisions and advance science. She serves as the USA NPN lead for key partnerships, including United States Forest Service, United States Fish and Wildlife Service, National Park Service, and Audubon. Coordinates activities to support the creation of new data projects and implements and oversees data collection campaigns at local to national scales. Erin oversees communications and works to establish new partnerships for the USA NPN. Erin received a BA in environmental biology from the University of Colorado at Boulder and an MS from the University of Arizona's School of Natural Resources and the Environment. With a background in wildlife conservation and management, she spent several years working as a wildlife field technician in locations across the country before serving in the Peace Corps in Vanuatu from 2007 to 2009. Erin has been with the USA NPN since 2010. She enjoys hiking, baking, and tending her small urban farm.
Thank you so much for that intro, and thanks for having me. Let me share my screen. So I'm going to be talking to you today about phenology and also sharing about volunteer science or citizen science, whatever you like to call it, and then talking more in depth about our Nature's Notebook program, which is part of the USA National Phenology Network. So thank you very much for being here on Valentine's Day. I'm happy to be spending it with you all. So I'm going to be going through kind of a definition of phenology and then talking about the many different applications of phenology for lots of different fields. I'm also going to be talking about some of the drivers or cues to phenology and the shifts that we've already seen. And then I'll go into the value of volunteer science before talking about the Nature's Notebook program. And then we'll do a little practice with some photographs of doing data collection in the style that we do it with Nature's Notebook. And then I'm going to talk about some of the projects that we have here in Arizona that we would love to have some help with. So to start off with, hopefully some of you have heard the word phenology. I feel like it's becoming more common, and hopefully your mind didn't go first to phrenology because that's quite a different thing. But phenology is simply the study of the timing of life cycle events of plants and animals and their relationship with climate and other abiotic factors. The word pheno comes from the Greek of to show or appear. You can think of kind of the difference between phenotype and genotype. Phenotype is the appearance of genes that can help remember what it means. And phenology is certainly not a new science. Indigenous people have been paying attention to changes in life cycle events of plants and animals since time immemorial. One example that we have of this is the different moons that you might have heard of that some people pay attention to. So things like the sturgeon moon or the beaver moon. Those are all related to what's happening in nature at that time of the year. And these things are pretty regular in when they appear, at least on a general basis. And so people have learned to be able to time different events or activities that they undertake based on what's going on a regular time frame in nature. And people have continued to use these different relationships with nature to plan different activities, including lots of planting of food plants. And so you might have heard some of these, but one I like is plant corn when oak leaves are the size of a mouse's ear. Once lilac flowers have faded, plant squash and cucumbers. And when crabapple and wild plum are at bud break, eastern tent caterpillars are hatching. So these things are generally depending on what's happening with the local temperatures and different things that are happening with the climate. And so that can help cue different activities that actually end up having kind of not only an interaction, but often just kind of a synchrony in the timing of their different events. I also like to share this quote from Aldo Leopold, one of our fathers of ecology, who shared that phenology, in short, is a horizontal science which transects all ordinary biological professions whoever sees the land as a whole is likely to have an interest in it. And I like this because it really demonstrates the cross-sectional nature of phenology. It touches on many different aspects of science, from ecology to ethnobotany, botany, and many different sciences that we study. Phenology is also a great indicator of climate change. And so even way back in 2007, the Intergovernmental Panel on Climate Change stated that phenology is perhaps the simplest process that we can track to pay attention to what's happening with species that are shifting in response to climate change. And it's something that anyone can observe. Anyone can go outside and start to pay attention to the changes happening in plants and animals and see evidence of climate change. It's also an early indicator of climate change as well as a simple process. So it's often something that will show up before we start to see decreases in population. So you might notice that a plant and pollinator are becoming mismatched before you start to see that that plant is not really reproducing and germinating and then having future generations. There are many, many different applications of phenology. Maybe one of the more obvious that I just mentioned is that of relationships between plants and animals, or animals and animals, or plants and plants. We can see this in different mismatches that are starting to occur between things like plant and pollinators, things like birds and their food resources. And another application is wildfire season. So understanding phenology can help to identify not only the timing of when wildfire season might begin, but also the potential severity. So knowing about whether there's a lot of plants greening up in the spring, followed by senescence, can indicate there'll be a high fuel load, depending on the timing of that senescence or the brown down of the plants and how that relates to the weather during that period, can indicate a time of high severity. Also, management of invasive species can be very informed by phenology. So here in Arizona, you all are very familiar with bufflegrass, I think. And that is a plant that can be managed based on its phenology. So knowing when it's green enough to spray with herbicide can help to know, and become more efficient with the timing of that spraying of the plants. Also agriculture, obviously knowing when to plant and harvest, but also knowing about when to treat for different pests can be, phenology can be useful for that. And also allergy season. So knowing about when flowers are open can be a more of an easy indicator of allergy season than some of the other ways that they try to track allergy season. Right now, I think a lot of places that try to estimate the amount of pollen in the air, it's a pretty intensive process where they actually have pollen counters up on hospitals, and someone has to go up and take down the sample and then look at it under a microscope. But by looking at when plants have open flowers and then how many flowers are open on the plant, that can help give a sense of when peak pollen release is happening. And we can understand not only the timing of the pollen season, but also the severity of when it's at its peak. And then also other pest management. There's lots of applications of phenology for treating forests and agricultural pests, knowing about when to, for example, put out traps for things like emerald ash borer or spray trees where adult insects are going to be emerging. So I would love to know if any of you pay attention or track activities of plants and animals. Maybe there's a favorite species that you have that you look forward to seeing, like maybe a butterfly or a bird that's migrating. Maybe there's a particular flower that you look forward to. But if you want to put in the chat just anything that you kind of pay attention to that's seasonal in nature, I'd love to hear about it. And I will open the chat too so I can see. But any favorite seasonal cycles? Lilacs in Michigan and spring wildflowers. Yeah, I don't know what it looks like up where you guys are, but it looks like we're going to have a pretty good wildflower year down here. We already have a lot of things coming up in our yard here. And I think not all of them are weeds. There are quite a few wildflowers. Argentinian cactus blossoms. Yeah, that's a good one. So feel free to keep putting things in the chat. Oh, saguaros. Now things are coming in. Pollinators and the flowers they come to. Monarchs. Yeah, and monarchs are an interesting one too that we have here at a funny time of the year. And then mesquite tree for cleanup, desert mistletoe. Hummingbirds nesting. That's great. So when we talk about phenology, if you think about that definition that I shared earlier, part of it is all about the relationships between the plant and animal life cycle stages and their timing with climate and other abiotic factors. So if you think about a deciduous, kind of typical deciduous tree, it's undergoing a lot of different changes over the course of the year. And it really needs to know when to undertake those changes to make sure that it's not, putting itself out there with fragile buds or flowers when the temperatures are going to turn. So plants and animals have really evolved to have a careful process for knowing when to undertake these different changes. So does anyone have any ideas about what might be the drivers or the cues for phenology? And you could think about our desert plants here or more typical temperate plants in other parts of the country or other parts of the world. But does anyone have any ideas of what kinds of things that have to do with the climate that might drive things? Seasonal rains. Yeah, if you think about our ocotillos here, it seems like they start popping leaves out within a couple days after a rain. Temperature and moisture for sure. And day length. Yeah, that's a good one too. Maybe not so much here for our desert plants, but in other parts of the country, for sure, day length or photoperiod plays a big role. So I think you guys got all the main ones that I had here, but those are definitely the key drivers, although there are some other things like nutrients and things like that that are kind of related to these as well. So one of these things, temperature, can actually be calculated in an interesting way called accumulated growing degree days. And this is a graph that we actually have in our visualization tool, and I'll be sharing how you can access that later on. But what we're trying to do here is show basically how much heat has accumulated in a particular place since the beginning of the year. And this can be calculated in different ways. You can have a different start date, you can have a different base temperature. But what we're using here is a January 1st start date. So basically from the beginning of the year and a 32-degree base temperature, which means any day where we got above 32 degrees, start adding up those degree days. So that would be, say it was 40 degrees on a particular day, that gives you 8 degrees. I guess it would be 7 because it's above 32, 7 degrees. And you add all those up across the year and you come up with the accumulated total. And a lot of plants and animals actually seem to, at least it seems to be, you seem to be able to identify when their transitions are going to happen based on how much heat is accumulated. They might be calculating it a different way inside themselves, but we could use accumulated growing degree days to kind of predict when different things might be happening. And often 32 degrees is used because it's freezing, and so a lot of things aren't really active when water is frozen. For some insects, it's actually more often about 50 degrees because that seems like the temperature where insects are more active above that. But anyway, so this graph is showing, I put in a location in Phoenix. And so we have a bunch of lines on here. You can ignore the green one. It's something funny happening with the visual right now, so you can ignore that one. The black line is an average of it's kind of our climatological average of 30 years, which is the period of 1991 to 2020. It's basically just the most recent 3 decades that have completed. So that's kind of our baseline, our average. And then we have 2023 on here as a yellow line, and then the blue line is 2024. And it's basically showing on the y-axis here is the amount of growing degree days that have accumulated, so how much heat. And so if we're under this black line, we're kind of behind the average, so we're colder, we haven't accumulated as much heat as that longer term average. And if we're under the yellow line, we're also colder than last year. So it's kind of interesting to see, I don't know if you can think back to January, but we did have some really cold temperatures back in there, and we've continued to stay kind of cool. And it's interesting to compare it to 2023 because it started out a little bit warmer and then it actually got even colder than we were back in the beginning of February. And then we have a little red line here that's the forecast for the next 6 days. But this, if you had a threshold in mind, like you're trying to get to 1,000 because you know that some activity, some life cycle event occurs when it gets to that point, you could see how close you are to that point and in how many days and kind of forecast forward. And so we have fun with this sometimes. So for Groundhog Day, we always do a little post, like an infographic kind of thing, and we compare our growing degree day map to what the groundhog has predicted, what Punxsutawney Phil said. So this map was as of February 2nd, and it's showing across the whole country whether locations are either cooler, they're like below that average line, or whether they're warmer or orange ahead in terms of heat accumulation. And so at the beginning of February, a lot of the southeast and the kind of southern Great Plains were pretty cool. They hadn't had a lot of accumulation, but a lot of areas along the East Coast were actually ahead. And then over here, we're just a kind of patchy mess. But here in Tucson and Phoenix, we were behind at that point. And then other areas like the Sacramento Valley area actually has been very warm. So that's unusually warm. I think one of the warmest years that we've had on record in the last 40 years. And then this is a white paper that is from a cooperative extension related office by Dan Herms. And he actually was able to calculate based on doing some experiments in a greenhouse, I believe, for a single location, how much heat has to accumulate for different plants to undergo a life cycle event, like a bloom, a full bloom, and egg hatch. And so this is actually the amount of growing degree days using a base temperature of 50 degrees before those life cycle events occur. And it's kind of neat to be able to have these. And there's still work that needs to be done to see if these are accurate for other locations. But it's a nice way to be able to have something that's fairly simple to calculate to reveal or predict when these different events might be happening. And then, of course, it's interesting to see, if you reach that threshold sooner in a particular year, is there a point when the plants or animals can't keeping up with the shifts and actually not doing the life cycle event at the time that was always predicted from the past? And then just a note on weather and climate. I really like this figure that kind of shows the difference between weather at a particular location, which might go up and down and have a lot of variability, versus climate, which is your really long-term look at what is happening with temperatures and precipitation. And then I like this phrase too, climate is what you expect and weather is what you get. And then this, I feel like, this is a graph of global temperatures for the month of January over the past 100+ years. And you can see that kind of variability, different Januaries can be quite different. And this is even, a global average, or sorry, US average. But even despite that, you can see, if we look at a long-term period, we can see a pattern emerging with the climate shifting to becoming quite a bit warmer, even though we might have some Januaries where it actually goes below the average and seems like it's quite cold. And then just thinking about putting this into thinking about from the perspective of a plant, for sugar maples, they might be impacted by weather in the sense that, they like to have cold nights and warm days for sugar to be, or for the sap to be flowing, and that results in great sugar production. But as things are warming, we're seeing a lot of increasing in nighttime temperatures. You might not get great syrup production in a particular year because of that. And then from the climate perspective, with those shifts happening year after year, you might actually see not only the decrease in the amount of syrup you're getting, but a decrease in the trees because they're no longer able to persist in that area because the climate is no longer suitable. So that's just kind of one example how you might think about the difference between the impact of climate versus the impact of weather. And then similarly with birds as well, you could think about something like a white-breasted nuthatch and how they might respond to weather that might be adverse in a particular year. If it's particularly cold, they might, shelter or cluster with other birds. If it's particularly hot, they might seek shelter in a cooler place. And then if they keep seeing that year after year, then that might actually shift ranges. And we're seeing many, many shifts in bird ranges, mostly northward or upslope to places that are more climatically suitable for these animals. And then in terms of the ability of animals and plants to shift, it can be quite different. So, think about plants might be able to shift in the timing of their phenology. They're not able to shift themselves physically to another location, except for by migration through different generations. But animals are able to move in place. But there can be other consequences too of the difference between these two types of organisms. And one really well-studied system is that of the pied flycatcher. This is a migratory bird that winters in northern Africa, and then it migrates up into Europe in the springtime. And this is a bird, as many birds that are closer to the equator, it relies on the day length or the photoperiod to know when it's time to start migration. It doesn't know what's going on, in the northern part of its range. There aren't a lot of changes in phenology happening at its winter location. And so it's kind of relying on just knowing, okay, it's time to go because the sun is telling me so, and it starts heading north. And typically they have kind of evolved to arrive at their summer nesting grounds at a time when they can have their nestlings come out of the eggs and they're ready for the abundance of caterpillars that will be there. But in recent decades, as things are shifting, it's becoming warmer earlier in the year. The plants that are actually hosting these caterpillars are able to shift. They're responding pretty well to these changes in climate. And so they're able to leaf out earlier. And so the caterpillars are also able to emerge earlier, and they're feeding on the leaves of the trees. But then the birds, because they're coming at the same time, are actually becoming too late. So by the time the nestlings have hatched, the abundance of caterpillars is gone. They're already turned into either pupa or adults at that point. So that food source is not there. And these researchers have also documented decreases in populations of the pied flycatcher. So they're seeing not only those early indicators of shifts and mismatches, but that trickling down to actually impact the numbers of these birds as well. Another example of a shift that's been documented is the timing of when understory plants are shifting versus the overstory deciduous trees. So in temperate systems where there's a lot of trees that are deciduous, they lose their leaves in the winter. That early spring period is a really important time for understory plants to take advantage of sunlight because it's the only time when there's a lot of sun coming down. And so that's when you'll see a lot of things like trillium and crocus and a lot of those early season plants. And so these researchers looked at the respective phenologies of the groups of plants on the forest floor versus the deciduous trees and found that the trees were able to take advantage of these earlier springs, but the understory plants were not. They weren't really keeping up. And so the trees were leafing out and blocking the understory plants. And they were seeing declines of these understory plants, just the numbers of those individuals over the years that they studied. Another example of phenological shifts that you might have heard of is that of the snowshoe hare. So the hares rely on camouflage with a snowy background, and with the loss of snow, earlier snow melt, the hares don't seem to be keeping up very well and changing back to their brown morph. So they are becoming kind of, I've heard it described as light bulbs on a dark background. And so these researchers studied the survival of these animals, and because they just stand out so much more for predators, and found that as the color contrast gets higher, the contrast of the hares with the background, their survival just plummets. So they're seeing decreases in those animals as well. Another interesting story of phenological shifts is from Kodiak Island. So the bears there typically will they feast on the salmon that are running and then laying their eggs and dying. And often they'll be it's kind of like a feeding frenzy. They'll be hauling out the fish and kind of squishing them, and then the eggs come out and they eat the eggs, and it ends up that there's all these fish carcasses all along the banks of the rivers there. And then once they're done feeding on the salmon, they transition and they go upslope and they start eating berries. They love elderberries. And then they, after they're done with the berries, they go into hibernation. And they found that as things have been shifting with temperatures, the elderberries are actually shifting. So they're fruiting earlier, and it's now coinciding, and it's at the same time as the salmon. And so you would think that the bears might choose salmon as the food source of choice, but they actually are choosing the elderberries, and they think it's probably because it's a high calorie food, and that's important for building fat stores. But they're missing out on the salmon food source. And the other consequence is the salmon aren't being hauled out onto the riverbanks as much. And so there's actually been some cascading effects of the lack of nutrients that typically were there from the decaying carcasses of the fish. And so there's other consequences happening along the banks where they're, the plant population is changing. And then there are many other examples of shifts that are happening across lots of different taxa, things like earlier leaf out and flowering of plants, earlier arrival of migratory birds and butterflies, earlier breeding and egg laying, and then earlier emergence from hibernation. That was one of the earliest examples that was documented of shifts in mammal phenology, was that of marmots in the Rocky Mountains. The way that things can shift can be really different as well. You could think of maybe a simple shift where this is maybe the full life cycle of a plant or animal. Let's say, for example, a plant that is flowering, you have the onset, the peak in when the most flowers are available, and then the end. And if it's a uniform shift, it all just happens earlier. And so that duration doesn't change. But in some species, we're seeing, like the first will change and then the last will change even more. And so you get actually a shortened duration. And in other cases, you might see that one end extends, and so you have a longer duration. So things like the amount of time that leaves are on a plant, or the length of the flowering period, which might seem good for the climate change perspective. If you have leaves on a tree for longer, they're taking up more carbon, which is a good thing. But it can be stressful for plants to have a shorter period of dormancy, and it can lead to, especially with drought conditions that can be there as well. There's a lot of stress that can happen, which can actually end up harming the plants. And then the autumn or fall phenology is much less studied than spring events, but I like to highlight this paper from a few years ago that looked at how things are shifting in the fall. And so for things like birds, it's really mixed what's going on. So some species, like the longer distance migrants, are advancing their timing when they're going back to their wintering grounds, whereas species that are shorter distance are delaying. Maybe they have more time, they don't have to go as far. For insects, some of the univoltine, the ones that only have one generation, are shifting earlier, whereas the multivoltine are shifting later. So there's a lot of interesting changes happening, and it's definitely still understudied. So there's a lot more work to be done there. All right, so now let's talk about some examples from the Southwest. So this was a study from, it's actually a long time ago now, but looking at broad-tailed hummingbirds and their migration timing. And so this is a bird that winters in Mexico. It comes up into the kind of southern Rocky Mountains in the springtime. And typically it has timed its migration so that it arrives when there's a lot of flowering plants available. But these researchers found that in recent years, the hummingbirds, by the time they arrive to their summer breeding grounds, the flowers have actually passed their flowering period. And so when the hummingbirds arrive, there's just not as much food available for them in those areas. Another study looked at the timing of flowering plants or desert plants, and then how that related to the timing of the winter monsoon. And back in the 1980s, the majority of the winter rains actually happened earlier than they do in recent years, and where they actually kind of peaked in October, November, whereas now it's more peaking in December, January. And so that actually has favored more cold adapted plants, because those rains are falling at a time when they're helpful to these more cold adapted plants. So that could be a potential shift to a totally different set of species that we're seeing in the desert. And then another example, so there's a gentleman, Dave Bertelsen, who some of you might have heard of. He's a naturalist who has been hiking Mount Kimball here in Tucson, which is a very steep hike. I've done it many times, and I'm really impressed that he's been doing it pretty much once a week for the last 30 years. And as he goes, he takes pictures of the plants. He calls them portraits. And then he also, he documents which species he sees at what elevations. And so he actually worked with the director of the USA National Phenology Network, Teresa Crimmins, to look at this long-term data set and see if things have shifted along this elevational gradient over the last couple of decades. And they found that after 25 years, 93 of the 363 plants had shifted upslope. And so they were seeing there are no plants that went down slope, but almost all of them that shifted were going up quite a bit to the more of the higher elevations. And then a more recent study from just a couple years ago, they looked at the timing of flowering in different desert species. And they saw over the couple decades that they looked at earlier flowering in all species, except for brittle bush, which was just kind of the same, that it had been a little bit earlier in recent years, but it wasn't significant. But all of these other species were seeing changes to earlier flowering. So if you want to know more about Southwest phenology, we have an info sheet that it's a little bit old at this point, but it does list some other examples of some studies. They're all long-term studies of 10 years or more that have found changes in Southwest species. So I can share that if people are interested. And then this issue of Madronio from a couple years ago was all about phenology, and it's open access. So if you just Google it, you should be able to find it. And that has some really interesting papers, including that one I just shared of the desert species shifting their flowering. So kind of in summary, Southwest phenology is quite complicated. There are a lot of things shifting, but in a lot of different ways. Some of the changes that are predicted for the Southwest we're supposed to get warmer on average. Precipitation is supposed to get more variable, kind of harder to predict. We could have more heavy rainfall, but overall it's predicted to be lower, although there's a lot of uncertainty around that. There may be longer growing seasons. I kind of wonder about longer mosquito seasons here. I feel like we already have such a long season, and I don't know if we'll get to the point where we just don't have a dormant period for mosquitoes, and they'll just be active all year round. Also more extreme heat events, although we still can get freezes, and there's been a lot of examples in the literature as well of or the consequences of having an early spring followed by a late freeze, and what that can do to plants that have already initiated activity. So the take home is we need more data. So that brings us to the topic of volunteer science. So we use the term volunteer science. I know that there's lots of different terms for this. A lot of people still like citizen science. I've had a lot of conversations with a lot of different people about this, including the person who claims to have invented the term citizen science, Rick Bonney, who's with Cornell Lab. And he feels like, you know, we've been working to get people to understand or become familiar with the term for so long, and citizen science is just finally catching on, and let's just go with it. He also feels like you can argue that we're not talking about US citizens or citizens of a particular country. It's just saying, you know, we can call them global citizens. So that's one argument. I've also heard people that like to use the term community science, which I feel like citizen science or volunteer science can be sometimes, but community science is actually also its own field, which it's supposed to be science that is led by and owned by communities. And often I feel like a lot of these crowdsourcing platforms are not community science. So I think it's a term to use carefully. I think when we're talking about, you know, groups of people that are coming together to do data collection and they're going to be looking at the data, using the data, definitely community science, but I think it's a term to use carefully. And then I recently saw someone was trying to get tracking science to be popular. I heard that a couple years ago, and honestly, I haven't heard it since. So I don't know if that's a term that is catching on at all. But I don't know if anyone wants to share their thoughts or preferences or what you've heard, if there are any other terms that you've heard. So the Citizen Science Association, which has been around now for a while, they actually have recently moved to change their name. And I can't actually remember. It's something to do with participatory science, but there's a couple other words in there, and it's become like an acronym. So I don't know how useful that will be for getting more people to get excited about it and involved in it, if it's kind of something that I can't even remember off the top of my head. But it's something that's still evolving. So it'll be interesting to see where it goes in future years. But we, at the USA National Phenology Network, we try to use volunteer science to distinguish the people that are doing this, that are unpaid, that are doing it as people who are not doing it as professionals, part of their jobs. And we actually engage with both volunteer and professional scientists in our data collection platform. So that's kind of how we distinguish that. Or we just like to use the word observers because that's what people are doing, and we like to call them our Nature's Notebook observers. So the history of volunteer or citizen science, there were many early naturalists that were untrained in sciences. A lot of early explorers could be considered volunteer scientists. And then the term was first entered into the dictionary in 2014. That's the term of citizen science. And then there was a great increase in these kind of programs in the 1990s as the internet took off, and it was much easier to host a platform that a lot of different people in a lot of different locations could participate in. There's several examples of the importance of people that are technically volunteers or citizen scientists that are tracking different events and things in nature. So Aldo Leopold, he documented the changes happening at the shack in Wisconsin, and he paid attention to things like the first arrivals of sandhill cranes and geese, as well as the timing of different plants. And his daughter, Nina Leopold Bradley, continued on that work over the course of many decades. And then she worked with several different researchers and put out this publication that was released in 1999. And they actually compared the observations from her father to the ones that she collected more recently, and found that over that 61-year period, the arrival timing of these birds actually shifted by 2 to 3 weeks, which is pretty significant in terms of the time frame. And we only are able to look at that because of those records that were kept decades ago. And then another example, there are many different naturalists who have collected and documented records. So Kathleen Anderson kept a careful record of when she saw different birds like wood ducks, and I think this was in Massachusetts. And so we can actually compare her data over many, many decades where she recorded the timing of when she saw these birds, and then compare it to the temperature and see an interesting change in the shifting date of arrival becoming earlier as the temperatures have been warming in her location. And then another example, gentleman Quackenbush did also keep records in a journal at a hunting lodge and collected observations of flowering plants, as well as different leaf out in trees and bird arrivals as well. And it's interesting that at this location, at least, the changes were not consistent. So the plants were shifting earlier for both leaf out and first flower, but the bird arrival was actually becoming later at his location. So interesting to be able to record that over a long-term period. In terms of the value of volunteer science, we can see this through a lot of different platforms, such as the Audubon Christmas Bird Count. That is one of the longest running volunteer science programs that is a national program. And so we have data going back to the 60s showing the bird abundance that's recorded through the Christmas Bird Count. And they've actually shown that the center of bird abundance, where the most birds are located in terms of latitude, has changed a lot in terms of miles. So this is a shift of over 40 miles that we've seen over the decade. So that is quite a big change. And it's interesting that we're able to see that because of those records that have been kept for so long. And this is over the whole country as well. Volunteer science has become pretty well used in the scientific literature as well. And this was actually just through 2014 that this study looked at, but they found that there was hardly any mention of any sort of volunteer science, participatory science, citizen science prior to the 2000s, but then it started increasing very rapidly. And now it's even higher, I'm sure. And the amount of people that are valuing citizen science or volunteer science and using it in research. And a couple studies that demonstrate this. One paper looked at the quality of data collected by volunteers and found that it was just as reliable as data collected by professionals. And then another study, this one actually looked at data from our platform, found that with limited training, the observations collected by observers were very reliable. And that's when they're following these protocols that we have that are very explicit and standardized, that really take all of the question out of it and just allow people to read a description and then answer questions. And this is an older study as well, but they found that volunteers contribute $2.5 billion annually to biodiversity research. So the impact that volunteers have is just huge when you look at the impact on biodiversity. So I'd love to hear now if any of you have participated in volunteer citizen science. Maybe there's a platform that you've participated in, but feel free to put in the chat if there's any platform that you've used. Oh, rain log. Yeah, we do rain log as well. We got a rain gauge 2 years ago and in 2021, and it was a great year for precip, and we felt like we had made it happen by getting our rain log rain gauge, and it rained for a bunch of days in a row after we put it out. Any other citizen science platforms or volunteer science? I'm sure that some of you have done one in particular. Yeah, like iNaturalist or eBird. Yeah, Christmas bird count, nice. Very cool. Okay. So there are many, many, many platforms out there, a lot of different programs to participate in. The one that I like to go to is SciStarter. A lot of you have probably heard of that. They just are like the main place to find programs, and you can find anything on there. Like if you want to do frogs or dragonflies, something in particular, there's a citizen science program out there for you, and they will tell you all about it. And so you can use their search feature. They have an advanced search feature too. So if you want to do something that's like definitely outside, or you want to do something that's only on the computer, there's just like a ton of different ways to participate. And you can say like how much time you have to give and all that, whether you need any equipment, the topical areas, and come up with a bunch of programs. And then this is a government website too, citizenscience.org, or government supported. And so they actually have a way to set up programs on here. They kind of have a platform to allow you to do that if you want to start a new one. I don't know if I recommend that because there's so many programs out there already. I feel like don't reinvent the wheel, just find one that you like, because there's probably already one out there that does what you want to do. In terms of phenology and volunteer science, there are many different programs that do this. So iNaturalist, I mentioned, helps you document anything you see basically in nature. You can take a picture of it. They have really powerful AI that allows you to do species identification. That's really exciting. And there's a great community on there too of people that will help you identify things and argue about it with you. Journey North is another one that does migrating species in particular, monarchs, hummingbirds, things like that. Budburst is another one that's more focused on younger grade levels. They do phenology observations in a little bit different way than we do with Nature's Notebook, but it's a great way, a very visual way to help younger kids document phenology. And then Nature's Notebook is our platform, which I'll be talking about a lot more next. And then there are probably some other ones too that I've not mentioned here, but SciStarter will help you find them if you're looking for something in particular. So I don't know how much time, should I keep going? Is this a good point to see if there are any questions? Because the next segment, I'll be talking about the MPN and Nature's Notebook, and we'll do some photo quizzes and things like that. But I'm happy to stop right now and see if there's any questions or comments or anything on what I've talked about so far.
Erin, I think this would be a good stopping point. We did have you start 5 minutes early, so we would be taking the bio break, but let's do questions. Dave, have you been finding any questions in the chat?
No questions.
All right. Well, I can give it a couple seconds in case anyone has one. And I see someone mentioned that SciStarter is a co-organizer for the City Nature Challenge. Yeah, which is great. Yeah, and I think iNaturalist is another great platform for doing BioBlitzes, right? Which is Nature Challenge is one of those. And then the Texas Pollinator BioBlitz and iNat. Cool.
And we do have a BioBlitz coming up also in April, so always good to know. Erin, I have a question. One of the slides when you were showing the rise and the increase of citizen science participation referenced something called SCOPUS, SCOPUS. What is that an acronym for?
Oh, that's a good question. Let me go back to that. See if I can figure out what. This one?
Yes.
I think this is one of those journal platforms. I can't remember which one, but it's like Web of Science is a consolidator of journal articles where you can search, and I think this is another one, but I can't remember what it stands for, but we could look it up.
Thank you. And I do see there's one question from Linda. Is there an Arizona Master Naturalist project on Nature's Notebook?
Yeah, so I'll get into talking about what we call our local phenology programs. So yeah, we have the ability for multiple people to come together and make observations at the same sites. And so we do have a Maricopa Master Naturalist one, right? I can't remember, is that the name of it? Maricopa? I'll have to look it up during the break, but
I think that's right. I think I set it up for one of our classes a while ago
yeah.
as like
Yeah,
a test. It was more testing stuff. I don't think there's anything like official in it, but
Right. So we can, if we want to use that, if someone wants to restart that or form a new one, we can use that if we want, or we could set up a new one. But yeah. And so, yeah, I'll be talking about the different ways to participate, but it's a great way to actually do observations together with a group of people and actually share and kind of like alternate who's collecting data. So yeah, we'll get into more of that after the break.
Well, why don't we take a 10-minute break? My clock shows 6:41, so if we regroup at 6:51, take your bio break now, everyone, and off we go with Erin when we return. Dave, you want to hit the record button again, please?
Great. All right, welcome back, everybody. So I'm going to share a little bit about the history of the National Phenology Network and some of our main goals for the network. So we were established back in 2007. There were a number of people that kind of recognized the importance of phenology for being able to document the impacts of climate change. Remember that IPCC report that came out in 2007. And it actually, our co-founders were a USGS ecologist and a professor who was at the University of Wisconsin in Milwaukee, who had for a long time studied lilacs and the phenology of lilacs. And so they kind of got together and said, what we need is a network that will operate across the whole country with the objective of collecting phenology data in a standardized way, storing those data, and then making sure the data are available for anyone who wants to use them. And so that's really remained our key objective for the network, is to collect, store, and share phenology data and information. We've kind of added on different aspects of what we do as well. Because of our historical funding with the U.S. Geological Survey, a lot of our objectives were to assist other federal agencies like the Fish and Wildlife Service, the Park Service, the BLM, in having the phenology information they need to make decisions. So knowing about the timing of migrating birds, or whether things were becoming mismatched, when to undertake actions to manage invasive species, and things like that. We also have always had a goal of advancing the field of phenology, so making those data available so that researchers can download them and then answer the questions they have about phenology in a lot of different fields and topical areas. We also have a major goal of wanting to make sure that phenology information is shared with the public, everyone. And so we do a lot of newsletters. We have a lot of campaigns where we invite people to collect data, but then we put a big emphasis on sharing the data back and making sure that people are becoming educated about phenology and they understand changes that are happening. And then more recently, we've seen a need to ensure that all of the data and the products and everything we do serves not just a small slice of the public, but everyone. So We are striving to create an equitable and inclusive network. One of the areas we're working on this is trying to do some Spanish translation of a lot of our resources to make sure that people that are Spanish speakers can contribute data, or at least use our education plans and learn about phenology, even if they don't collect data. And then we're also involved in things like the Indigenous Phenology Network, which is a national effort to connect people, both Indigenous people and non-Indigenous people who care about phenology. And they have a great set of monthly calls that people are invited to join. So I can share more information about that if anyone's interested. So the primary way that we do this data collection and storage is through our Nature's Notebook platform. This was started pretty soon after we were established. It started going in 2008, but really 2009 was when we had it widely available. And now we have, I think, close to 35 million records in the National Phenology Database. And this map shows kind of the density of records coming in from the different sites across the country. So each of the little gray points is a data collection site, and then the circles represent places that have collected a lot of records. So, the biggest kind of magenta circles have collected 600,000 or more records. A lot of those locations are sites that belong to the National Ecological Observatory Network, which some of you might be familiar with. That is a national network. It was started through a National Science Foundation grant, and it more recently is overseen by Battelle. But they use our Nature's Notebook protocols for plants, and they do really frequent surveys of plants at their neon sites, which I think there are about 50 across the country. And we import those data into our database as well. And then you'll see other areas where we have a lot of records coming in, a lot of the urban centers, but a lot of our local phenology programs, which I'll talk about later too, are responsible for bringing in a lot of data into the database, a lot of important records. And then you'll see some gaps. We have a lot of gaps in like the Great Plains state, so in some parts of the Southeast. So we're always trying to get more observers in those locations. So there are two different ways that we have for participating in Nature's Notebook. One is to participate as what we call a backyard observer. You can do this independently in your own backyard or another place that you go to frequently that's convenient to you. Or you can participate as part of a local phenology program. So I'll share more details about that, but basically it's when you're coming together with a group of people, you have a shared goal, maybe species that you're all interested in, or you have a question that you're trying to answer, or maybe it's more of an education or outreach objective where you're trying to engage people, a group of people in learning about phenology. And so you come together and you actually share the same sites where you do the data collection. And we found that people in these different groups have kind of different motivations for participating. A lot of our backyard observers, one of their major reasons for participating is contributing to a national effort or contributing to science. A lot of them feel like they learn a lot, and a lot of them are interested in nature. And some people say that it gives them a chance to slow down, spend some time outside, and really get to know the plants or animals that they're studying. So you'll find, after you look at the same individual plant over and over once a week for a year, you really start to notice like little details about it that you wouldn't have seen otherwise. You'll start to see the first tiny little buds that start emerging, especially with some of our desert plants. They don't often even have bud scale, so it just seems like all of a sudden there's a leaf popping out of the side of a branch. But you'll start to see those first little signs of that. Or you'll notice when a flower goes from being closed to open, and you can see that change happening within a day. So there's a lot of really neat things that you end up noticing and kind of little details that you start to pay attention to. With our local phenology programs, generally people are either coming to this because they have a science question they want to answer. They might be concerned about, mismatch between a plant and a pollinator, or the timing of something shifting. And so they have a specific question or set of questions to answer. They might also have a management goal. So it might be, we want to study this species of invasive grass, so we know exactly when we're going to get out there to treat it at our location, or know when to remove it so we can avoid before it goes to seed and start spreading seeds. And then some have a more of an outreach or education objective. Like I mentioned, it might be, we want to engage visitors coming to our nature center. So we're going to have this as an activity, and we're going to do guided walks, and we're going to have people participate in Nature's Notebook as we go around our trail or something like that. So the way that we record phenology data is different for animals and plants. So with animals, you're doing this at the site level, site, which is your location, and you're reporting on the activity that you see, any reproductive behaviors, and then developmental stages. So things like when you see nestlings in a nest or fledged young. For plants, it's a little different. So we actually have you report on the same individual plants over time. So you would find an individual saguaro or a milkweed plant, or maybe a patch of plants. If you're not really sure if it's kind of a mess of a few different individuals, you can mark it as a patch. But you're going back to that same location, that same individual plant over and over again. And for plants, you're recording what's happening with the leaves, the flowers, and the fruits. And it's pretty powerful once you start doing that, because you can start to see not only the changes across the seasons, but you can see the changes between years. So you have a saguaro, it might start flowering a month earlier one year than another year. And that's really valuable information to have, because it's showing that this same individual plant is responding to the conditions of its environment, and it's demonstrating that through the appearance of its phenology. So a lot of the kind of journals and things I mentioned earlier by those early naturalists were what we would consider to be event monitoring, which is a type of phenology data collection. And that's often when you record like the first appearance of a migrating bird, or the first time you see flowers on a plant. And that can be useful. You know, you can look at events over time and see the changes. Maybe it's an earlier event this year than last year. But it's hard for a scientist who's using your data to know that that's reliably the first event, because they don't know if you missed it by a week. They don't know if you happened to go out in the middle of the summer and say, oh, this is the first time I saw this flowering plant, but really it had started, a month before. So the kind of monitoring we do is called status monitoring, where you're going back to the same plants. And every time you visit, you're going to be reporting either yes or no. And those no records, the negative data, are really important because they allow us to pinpoint when the first yeses start. And then they also allow us to see if there's multiple periods of activity. So you can not only pinpoint the beginning and the end with a pretty precise estimate, but you can also see maybe there's, multiple flowering periods, which happens on a lot of our desert plants, like things like creosote, where they might flower one time and then they stop and then they flower another time. And if you had only recorded the first event, you would totally miss out on all that other activity. And we actually do one additional step, which is we also have abundance and intensity data that we ask our observers to collect. And so not only are you saying, I saw open flowers on my creosote, but you're trying to estimate about how many flowers you saw, how many flowers, and then what % were open. And so what that gives us is not only the presence absence of a phenology phase, but the peak or the magnitude. So you can see, maybe there were flowers twice in a year, but this first flowering period was much larger. And you can see that in things like saguaros. There might be a fall flowering, a second flowering in saguaros, but it's much smaller and usually only a few flowers on the plant. Whereas in the springtime, that's when the majority of the flowers, the majority of the nectar and the pollen and all that is available. So that's really useful information to have. And this is useful for a lot of different cases too, not only with flowering, nectar and pollen, but also fruiting of plants as a wildlife resource. And for animals, when the peak in the migration is happening, or the peak in amount of birds nesting. So here is a photo, and I would just love it if anyone wants to put in the chat, just give me some descriptions of what you're seeing here. It can be whatever you like, however amount of detail you like, but if you just want to jot down a few things about what you're seeing, what you're observing in this photo. And it's totally open as to what you say. Okay. Seeing leafing and flowering phenophases simultaneously. Yes. Anything else in there? Any other? There's probably a lot of people writing a long paragraph right now. Okay, pine needle milkweed flowering and monarch caterpillar. Getting a little more specific, monarch caterpillar. Okay. Got some species information in there. Yeah, I'm gonna give it another couple seconds in case anyone else is typing. An assassin bug in the background. Wow, that's keen eyes. I don't know. It's possible. I think it's also a little blurry, which is my fault. I think I focused on the flowers and didn't get the rest of it in focus. I saw the bug. That does not appear to be a place the caterpillar should be. It looks like, yeah, you might have eaten off all the leaves and gotten stuck there. Seed bug. So, okay, so my point of doing this is there's a lot of different ways that you could describe what's going on in this photograph. And so this just kind of drives home the point of why we have standardized protocols, because it allows you to look at something like this and not just a photograph, but in real life, look at a plant and record it in the same way that everyone else does. So you can more easily compare data across plants, across species, across areas. And it's just much better for data analysis. So this is an example of one of our protocols, or the set of questions we have as our data sheet for a particular species. This one is for desert or rush desert milkweed, Asclepias subulata. And we actually have protocols for over 1,800 different species now. I think there's at least several hundred, I want to say 500 in Arizona. We have a lot of different species that we've added. We are limited in Nature's Notebook to only observing the species that we have on the list, but we do have a process for adding species, especially for our local phenology programs, because we love them. We will add species on request. So if you start a program and you realize, oh, the species we really want to track is not on the list, we can add it. It'll just take a little while. We only do it once a year. And we're just doing it right now. So we take requests up through August, the end of August, and then we add them the following year, and usually the end of January or February. So that's kind of the process for that. So this data sheet for the desert milkweed has a few different questions here. We have our leaves questions in the dark green. We have flowers questions and fruits. And you'll see that for each of these, we have a Y for yes, an N for no, or a question mark. And so each time you go out to observe, everything's optional. You don't have to complete the entire data sheet, but we do like it when people fill out the whole thing. And we really like it when they make sure they fill in the no's, because those negative records are just so important, like, as I mentioned, for being able to know when something started or stopped. So every time you go out, you could complete the entire data sheet, answer yes, no, or not sure. We also have this blank here, and that's for that intensity or the abundance. And so for each of the questions on the data sheet, we give you a definition called our phenophase definition. And what we're trying to do here is we're trying to take out any of the guesswork that might be associated with trying to know, for someone who's not a botanist, well, what does an open flower mean? How do I know it's actually open versus just any flower? And so we tell you here that flowers are considered open when the reproductive parts are visible between or within unfolded or open flower parts, and then don't include dried or wilted flowers. And so for things like fruits too, we actually will give you a species specific definition where we're telling you for this plant, you'll know that it's a fruit because it's large and pod-like and changes from green to tan or brown. And so we'll also tell you for ripe fruits what it looks like when it's actually ripe. And so we're trying to give you all the information you need as a non-expert to be able to answer these questions in a standardized way. And so with the abundance data too, we have a question here, how many flowers or flower buds are present? And we're not asking you to count how many actual flowers are there. We're just having you put your estimate into a bin. And so they're kind of like exponentially increasing bins for our numbers. So less than 3, 3 to 10, 11 to 100, and so on. So this can be daunting at first, and we definitely recommend if you don't feel comfortable with this, just skip it, just answer the yes, no, and then move on. You can always come back later, and start adding that into your observations. But once you start ruling out bins, I feel like it becomes much easier. So usually when you're looking at a plant, you can just by eyeballing narrow it down to 2 of them right away. And then you're just trying to figure out which one does your estimate belong in. So I would just say, don't overthink that part of it. Do your best. And if you don't feel comfortable, just skip it. For the percentage questions, we have bins that are basically quartiles with little tails. So we have less than 5%, 95% or more, and then the rest of it's kind of split up into quartiles. So this is just meant to help us estimate the peak in when things are happening. So again, it's not super critical, if you get less than 5 or 5 to 25%, if you're going back week after week and you're answering the questions and you can see that progression over time and you can identify the peak in when the most were on your plant, that's really what we're looking for. And it definitely becomes easier over time. And as you're going back to your same plant week after week, you know what you said last time, and then it's pretty easy to say, oh, there are a lot more this time. I'm going to move my estimate up and judge it that way. Here is one of our protocols for monarchs, which is, you'll see it has a lot more questions on it. We have a lot of questions for insects as well as birds. We kind of restructured our protocols after working with some of the Audubon chapters in California who are using our bird protocols, and they really wanted to capture a lot more of the life cycle stages than we had originally. So we added on like a lot more of the stages for insects and birds. So again, we're trying to give you, definitions of what these things look like. And so it's a little bit more obvious for some of the animals, but for something like eggs, it'll give you a description. These are tiny ribbed, dome-shaped eggs. And then for caterpillars, it'll tell you what exactly the caterpillars look like. So again, you're answering yes or no questions or question mark. And then with the abundance for these, for animals, it's simply a number of how many you estimate that you see in that phase. There are a couple different options for data entry. We recommend using the app. We actually have been trying to make the app better. There's still some issues sometimes with it, but you can now do pretty much everything in terms of getting your account set up, setting up a site and plants and animals on the app. The only thing you can't do is add more of the kind of metadata, the details about your plants that you still have to do on the website. But it's really set up as kind of a mobile data sheet. So if you want to get everything set up on the computer and then use the app as kind of your data sheet in the field, that's fine too. The app works offline, and then the data are stored on your phone. Just make sure when you get back into Wi-Fi, you open it up again and even go to this on the review menu, there's a sync button just to make sure that your data are pushed to the database is always a good idea. But that does work offline. And then we do have paper data sheets too. If you prefer that, you can print them from the website. And then we have this data entry interface that looks very similar to the data sheets that you could just transfer your data on there. Okay, so I haven't numbered these photos, so I thought we could go through and try to answer them. We don't have polls set up, but I think we could do this with the chat. We're gonna try it. Or you could keep notes to yourself if you want. But let's go through and for this photo here, and hopefully you can see it. I'm not covering it up. I don't know if my chat window covers this for you all, or if you can see it just fine. But this is a data sheet for a milkweed, which all the milkweed data sheets are pretty similar. And so we have some questions about initial growth, young leaves, flowers and flower buds, open flowers, fruits, and ripe fruits. So we're just going to do a couple of these, the ones that we're kind of focusing on for our desert refuge project. So let's have everyone take a look at this. So we're going to do the question of, do you see leaves? And I've posted the phenoface definition in white here. So everyone take a look. I know it's kind of hard because the picture is zoomed in and it's kind of cropped, but do your best. And would you answer yes, no, or question mark to this question of, do you see leaves for this photo? And we'll see if we can get a consensus with a few chats in the chat.
You see leaves? Yes.
Yes. Okay. We're getting a lot of yeses. That's great. Okay. Let's go to the next one, which is flowers or flower buds. So do you see flowers or flower buds? One or more fresh, open, or unopened flowers or flower buds are visible. Yes, we're getting yeses. All right, nice job. That's a yes too. Those are flowers. And so for this one, it's really asking about any flowering activity. So if all of these were buds, it would be a yes. If all of them were open, it would still be a yes. And then really it's supposed to be an overarching phenophase. So if you say yes to open flowers, you have to have said yes to flowers or flower buds. But you can say yes for flowers or flower buds, but not say yes to open flowers. So flowers or flower buds is like any unopened open flowers. And it says that right in the definition to help you remember. It's any flowers.
Thanks.
Nice job. Okay, and now the last one for this is open flowers. Do you see open flowers which have the reproductive parts visible? Which is kind of a little blurry now. I'm noticing when I zoomed in on the photo.
Love you.
Yes, we're saying yes, we see open flowers. Milkweeds are really cool.
So good. I love you.
Okay. So now let's move on to the animal side of this. So this is a data sheet for a monarch butterfly. So do you see adults in this photo? One or more adults are seen or heard. Probably wouldn't hear them moving
love-
about. Or...
Oh, good boy. He's so good. He's so good. He's so good.
I think someone might be talking to someone who's down.
Yeah, somebody's not muted, just FYI.
Okay, so we're getting a lot of yeses. That's great. Yes, there's a big monarch in this photo. Nice job. Okay, how about mating? A male and a female are seen coupled in a mating position. And I picked a few of these because these are the ones that we're interested in for our desert refuge campaign, which is one of our data collection campaigns. But nope. Probably not. We only see one butterfly here, so we're going to say no for that. And important to report the no. How about egg laying? A female is seen laying eggs on a plant. I see a question mark. I like that. It's a little hard to know. I mean, it's possible, but I would say it's not visible in this photo, but it's possible. And of course, if we were there in person, it might be a little bit more clear. But I think a question mark is a safe option for this one. A nice job with that. Okay, and then the last one, caterpillars. You see a caterpillar, and it tells you for monarchs, young caterpillars are pale green with a black head. Older caterpillars have 2 pairs of black tentacles and black, yellow, and white stripes around their body. No, yeah, I don't see a caterpillar either. If we were able to search the whole plant, maybe we would find one, but I don't see one in this photo. So I think no is the best choice. So nice job. Okay, here's another one. So this again, this first data sheet is our milkweed data sheet. And this again is kind of zoomed in, so question marks are welcome, but do you see leaves, which is one or more live, fully unfolded leaves visible on the plant? Yeah, most people are saying no. I would say no as well. There might be some leaves somewhere in the fuzzy background, like, I don't know, maybe that, but that could also be a stem. So I think no is a very safe bet for this one. How about flowers or flower buds? Yes. One of them's getting munched. Yes, consensus is yes. How about open flowers? Okay, mostly yeses. I see a no in there, but mostly yes. Yes, I would say yes. These are definitely open. These are not open, but these are open. Some of these might be getting close to the dried stage, but these ones look kind of fresh to me, and there's lots of bugs on them. So it's a good sign that they're open and fresh. Okay, how about this is the monarch data sheet again. So do you see any monarch adults? No's, lots of no's. Okay. What about mating? You'll learn that you report a lot of no's for things in Nature's Notebook. A lot of our records are negative records, which are still important. And we do on the app have an option and on the website to circle all no, which I always do first, and then I go back and put my yeses in, because more often than not, it's like the majority are no's and then there are a few yeses. Okay, so no for mating. What about egg laying? No, okay. And then what about caterpillars? And pay attention to the description here, because I'm trying to trick you on this one a little bit here. Someone's on to me. Linda knows. So this is indeed a caterpillar, but it's not a monarch caterpillar. This is a queen caterpillar, and we can know this if we look at the description. We don't really have a sense of how big this caterpillar is, but older caterpillars, this is a pretty big one, have 2 pairs of black tentacles and black, yellow, and white stripes around their body. And so one of the main differences between queen and monarch, they're in the same genus, so they are related, is the numbers of sets of these. They're like tubercles or tentacles. The monarchs only have 2 sets. So they have one at the front, one at the back, and the queens have an extra set here in the middle. And they have this kind of like reddish purpley color that monarchs don't have. And the stripes are a little bit different. So this is a trick, not a monarch. So you would say no for monarch caterpillars. But if you also had queens at your site, you could say yes for queen for caterpillars. Okay, so last one. So again, this is a milkweed data sheet for this milkweed here. So do you see leaves? Yes. Okay, we're getting a lot of yeses. Yes, there's definitely leaves here. I didn't ask about these other ones, the young leaves. It's kind of hard to see because there are some young leaves in this photo, but they're just blurry. So I didn't include that one. How about flowers or flower buds? At least in the photo. No, yeah, no flowers or flower buds that I see. And what does that mean for, does anyone know what our answer for open flowers will be immediately if we said no for flowers or flower buds? No, yeah. If we say no for this one, we have to say no for this one because you can't have open flowers without any flowers on the plant. Okay, how about for monarchs? Do you see any adult monarchs? Nope. Yes. Someone saw one. I don't know where it is, but I would say no. I do not see any adult monarchs there. How about mating? It's getting a little repetitive. You'll see that as you do this over and over, week after week, it gets really quick because you'll already know the next question on the data sheet and you'll just kind of breeze through it. Okay, no mating. What about egg laying? No. Okay, what about the most exciting one for this photo? Is this monarch caterpillar? No one, yes. Anyone else? I might have scared you with the last one. And it is a little hard to see because you can't see all of the middle, but yes, this is a monarch caterpillar. So we see the colors are right. We got black, white, and yellow stripes. There's no reddish color. And it's a little, this is kind of misleading, but there's only 2 sets of tentacles on the front and on the back. There's nothing here in the middle. it's a black tentacle. So good job. All right, so I wanted to share some of the other training resources that we have available. So when you create a Nature's Notebook account, you'll have what's called your observation deck. This is the place where when you log in, it's the place where you access all of your resources. You can set up all of your sites and plants and animals there if you want, if you don't want to do it on the app. We also have, I mentioned, the place to add in more details. So if you want to spend some more time and give us some of that rich metadata that we like, that not a lot of people enter, you can put more stuff about your plants and the sites that you're setting up. So for example, you could say, I'm registering a milkweed plant and it's in a shady area and I planted it on this date, if you know that, and it does get supplemental water, but it doesn't get fertilizer. And all that stuff can really help us to know what's going on with like other conditions that are influencing the phenology of your plant. So definitely check that out if you have the time and interest. We'd love for you to report that information. And then for your sites, you can put things like, do you have a feeding station for birds? Like, do you have hummingbird feeders out that might draw in birds? Or are you close to a street or a stream, or is your site urban or rural? And it'll ask you a bunch of questions like that. There's also a learning tab on your observation deck. I'm sorry, this is kind of blurry. I don't know what happened to the image, but there's a number of resources on here, including the botany primer, which I know that you all have gotten a link to already or will. This is our 80-some page guide that is a very photograph-rich guide all about botany that is useful for using Nature's Notebook. We also have an observer certification course, which I think I have linked here. So this is there's 5 modules that are part of this course. It's optional, but definitely recommended, especially the first module. So the first module is just the basics on how to use Nature's Notebook, how to set up a site, add plants, animals, how to enter data, edit data if you need to fix something, and then some kind of tips on how to pick out a site, how many plants to choose, where to pick your plants and all that. We have a second module that's all about the mobile app and how to use that. The third module goes into a little bit more depth on our phenophases, the questions that we have on the data sheet and kind of why we selected those and how the data are used. And then our fourth module is all about estimating intensity, which is one of the more tricky things, especially if you're observing something like a mesquite tree, where, it's really big sometimes, and there's a lot of flowers and a lot of seed pods. And it'll give you some tips on how to do estimations with some tools. And then the last module is just like a practice where you watch a video and then you answer some questions. So the whole thing we estimate takes about 5 hours or so. You don't have to do all of it, just the first one is great, but it's there if you want more detail. We also have these definitions, which I was sharing earlier. That's the description of exactly what we're talking about for the life cycle stage. Those are all available on the website. You can print them out and also on the app. So when you answer a question, you'll see a little eye next to it. If you tap that, it'll bring up that phenophase definition. And then the botany primer is what's linked from the observation deck. I also want to mention we have a number of campaigns that we would encourage you to join. So these are focused data collection efforts. They're usually initiated by researchers who come to the NPN and they ask us, can you please ask your observers to collect data on these specific species? I really need the data for my research. Or it's people like the U.S. Fish and Wildlife Service that want data on things like lesser long-nosed bats, or their food resources at least, so they can kind of track them as they've been taken off the endangered species list. We have a lot of these different campaigns, and you can see all the different ones on our website and read the description. The ones that are most relevant, I'm going to be talking about those as ones that need your help. But some of these are national too, like Green Wave is about maples, oaks, and poplars. We do have some cottonwoods in places like Sabino Canyon here. So I don't know if you all have places with cottonwoods up there, but you might find something on that list. Nectar Connectors includes all the milkweeds, and so that's a relevant one as well. If you have any oak trees, these are mostly eastern oaks, but there might be one, especially if you're near like a botanic garden or in a place where they planted street trees. Sometimes there's some oaks on that list. And then Pest Patrol is another one. We don't have a lot of these pests, but there are some that are in Arizona. And then Pollen Trackers is also a national campaign that it includes a lot of allergenic species like oaks and maples and birches and junipers and things like that. And then redbuds, actually, you might have redbuds there too. We have, they're actually, I guess they're either Mexican or Eastern redbuds. We have a mix on campus here in Tucson, so that could be another one too. But what the campaigns are basically you're signing up for a series of newsletters from us. And so we'll send out information about why we're interested in data from these campaigns. We'll send out lots of results of who's reporting what. We'll send you maps of where the data are coming in from. This is the Flowers for Bats campaign. This is focused on columnar cacti, saguaro, cardone, organ pipe cactus, and then agaves, palmeri and perrii and desert eye. We do have Americana on the list. It's not really one that the bats use, but we don't know what's going to happen with the future and climate change. So and a lot of people have it in their yard. So we're including Americana on there too. And you can see the species list on the website. But we share back identification tips and reminders of when to start looking for different life cycle stages, lots and lots of these kind of results of what people are reporting. Here's another one, nectar connectors. That's the one that includes, it's actually about 54 different species that are all across the country. We kind of pick those because they're important for monarchs, but they're also important pollinator plants for a lot of different species. This is a national campaign, so you'll see people are reporting all across the country. And we have some other things too, asters and sunflowers and lupin and things like that, in addition to the milkweeds on this list. We also have a way to download data. If you're interested in downloading data sets, you can get basically an Excel file from our Phenology observation portal, and you can filter the data. If you're interested in data on particular species or locations, you can see what we have. And then, there's other ways to filter as well. The partner groups is where you can add things like the Maricopa Master Naturalist local phenology program if you only wanted to download data from that. And there's some other ways you can do that too. Once you have a program, we put the link in the chat, but it's kind of the profile page that has a bunch of resources. I can show that at the end too. But this is kind of where we send people that want to download data sets. If anyone works in R, we have an R package as well. And we have a number of vignettes that kind of describe like data cleaning procedures and how to work with the intensity data. So if anyone's an R user, I can share the CRAN package link as well. This is just a couple examples of some papers that have used the data. I think it's a really important part of what we do is making sure that all the data that are collected are made freely available for scientists to download and use, scientists as well as anyone else who wants to use them. But they do get used, and we have had, I think we're up to about 130 peer-reviewed publications that use Nature's Notebook data. So you don't know for sure that your data point will be used by scientists, but it's going to be out there, and we've had lots of people use data. So you can feel good that the data you submit will probably be used by someone at some point. And our data sets have also, I should update this, now Phenology and the Nature's Notebook data are in the 5th National Climate Assessment that just came out. And it's also in things like the EPA's indicator report. I just wanted to talk about a couple of the applications that have been that have used Nature's Notebook data. So there's a lot of different ways, but some interesting ones are selecting species for restoration. So we have a project that's in the south central part of the country right now called Time to Restore. And that effort is focused on helping people that are doing pollinator restoration know what to plant for future climate conditions. So we're trying to figure out when things are flowering now, what are the climate drivers, like we talked about earlier, to those plants, and then use climate projection. So say if we know temperature and flowering in this particular species are really closely related, and we know that with every degree increase in temperature, this plant is going to flower 2 days earlier. We can project forward and say, well, we think that this plant is going to flower maybe like 10 days earlier because it's going to be this many degrees warmer by 2050. And that gives people a sense of when things might be happening under future climate conditions. And so if they want to, for example, provide bloom coverage for different pollinators during like the spring, summer, and fall, they want to be able to take that into account and know those changes that are going to be happening in the flowering timing. It also helps in timing management actions. I talked about the buffalo grass example. We've also had other projects that have looked at other invasive species to know when is the best time between when the plant is leafing out and when it seeds. So I can make sure that I send out my crews to take out the plant before it goes to seed. And then I have like a whole nother generation of plants to worry about. Also risk assessment. We've had some interesting papers to do with fire risk and looking at for a plant called chamise in California, knowing the flowering status can actually inform when or how flammable the plant is when it's most flammable. And so without having to use complicated tools to assess like the moisture level in the plant, they can just look at whether or not it's done flowering, and then they know that that means it's the most flammable and they should take extra precautions. And then I think the last one, I do have an example here. So the McDowell Sonoran Conservancy is one of our local phenology programs, and they have been interested in the interaction between saguaros and white-winged doves. And so that's those are a couple of the species that they've been tracking for many years now. I think they've been with us for, I think, 8 years. So this is one of our activity curves, one of our visualizations from our Viz tool that shows when people saw white-winged doves and then when they saw saguaro open flowers. And so you can see kind of the interaction between those two species from the data they collected in that year, and see that the doves actually arrived earlier than the open flowers were available on the plants at their location. And then there are many different science applications as well as management applications. So just understanding when things are happening and kind of what are the drivers to different phenological events. There's been a lot of work done with our data on Southwest species in California. They act really different compared to the typical kind of species up in the temperate areas where it's mostly photoperiod and temperature as a driver. So they've been looking at precipitation, snow melt, things like that. And then there have been quite a few papers that have started to look at the changes over time, trends that they're seeing in things actually shifting in addition to just the relationship between them, and then projecting forward how things are going to change in the future. And one example of this in particular, this was another Nature's Notebook campaign that we ran for multiple years, a couple years ago, called Shady Invaders. And this was actually a campaign initiated by a PhD student at Penn State. And she really wanted data on shrubs in the eastern part of the US that are invasive and native shrubs. And she wanted to test this hypothesis about this extended leaf phenology. It's a hypothesis that invasive species, they're better adapted to new environments. They're able to like colonize new places. They're probably also better able to take advantage of earlier temperatures. And so she compared the phenology, the leaf out of invasive shrubs to native shrubs across the country. And she found that especially at lower latitudes, there's a really big difference. And invasive shrubs leafed out way earlier, on the scale of months earlier than the native shrubs. And so they're able to take advantage of that period. Remember, when the deciduous trees don't have leaves and there's that sunlight coming into the forest floor, the invasives are really doing well in the early springs, but the natives are just like starting to leaf out at that time. This is less pronounced at northern latitudes, but it was a really interesting finding after just 3 years of data collection with Nature's Notebook. So lots of other examples of how different researchers have used our data. We keep track of these. We try to publish at least like every other month a summary of one of these research studies, and we put them out in our newsletters. We put them on our website at this link here. And I'll share a QR code for our newsletter sign up at the end. But if you're interested, it's one of our goals of communicating and connecting about phenology is just to try to make sure people are aware of the most recent science. So there are a lot of publications out there, but we try to do one every other month. Also, if you want to play around with the data, you can try the visualization tool. So this is the link for that here. When you first get into the tool, there are a series of these little panels that we call seasonal stories. And that will kind of give you an overview of all the different types of visualizations. It'll have one set up for you and tell you what the tool is showing and then how to manipulate it to look at different species or years. We have tools like a scatter plot tool that allows you to compare the phenology to climate variables or other abiotic variables like latitude. So you can look at, for example, this is all maples across the country. And when the leaves are first coming out, you can see at southern latitudes, it's much earlier than at northern latitudes, which is what we would expect. We also have this tool here, the one that I showed earlier with the accumulated growing degree days in Phoenix. So you can go on here and put in your own zip code and kind of track that over the course of the spring and see if we catch up to normal within the next couple of weeks, or maybe we'll probably get past it. Honestly, we'll start out slow and then it'll probably get really hot in March, and then we'll be ahead of our long-term average curve. And then another activity curve here, just showing again some of the data that you can look at. So you can look at multiple phenophases of different plants. You can compare plants and animals. You can also compare different years. So you could stack up a bunch of years and see how the consistency is, or if it's variable between years. So for example, with 2021, we saw the white-winged dove were arriving just kind of right before the open flowers, and then there are fruits that the doves could feed on as well. And then this one here from just last year, the doves seemed to arrive, I guess comparing the two, there was kind of similar arrival, a little bit of a bump earlier, but the flowers seemed to be a bit later this last year than a couple years ago. Okay, so just a little bit on local phenology programs. So I mentioned these are when you get groups of people together to observe phenology together. And I just wanted to mention we have a ton of resources available for these different groups. We have a certification course, and I have some more information about that later. For local phenology leaders, if you want to get certified as a local phenology leader, you can do that. We have a ton of program planning resources that are available to anyone who creates a program. We also have a lot of helpful materials on volunteer recruitment and retention. This is particularly through our community of practice. So we have a monthly call that we do on the third Friday of the month, and anyone is invited to come. We usually do like a share out of what's going on with different programs. There's a chance to ask questions. We have at least one of us as a staff person on the call. So you can ask us about like what's going on with my data, or I'm having trouble like troubleshooting this issue, and get all your questions answered. We also have a listserv, which is a Google group. We have a Facebook group, and we have a newsletter that goes out to local phenology programs. So who contributes? This is kind of an older side too. We have a lot more recent partners too, but this just kind of shows the diversity of different partners that we have across the country. All the little gray dots are personal sites or backyard sites, and the little green ones are local phenology programs. So you can see it's kind of like evenly spaced as across the two types of participation. We do ask people when they start a local phenology program what their reason is, what they're hoping to do with their programs. And by far, the most people are studying climate change impacts or answering their own science questions. But we also have people that are just trying to engage, visitors, the public, in doing citizen science, getting students involved with research, just getting a baseline on what's going on with plants and animals. And then if you look at the breakdown of the types of organizations that participate, a lot of botanical gardens or arboretums, we have a lot of university classrooms or K through 12 schools. It's kind of a neat way to engage students that might be moving on after a year, but still have a data set that remains. So the teacher that's there can see how things are changing from year to year and share back with the new students what previous students have reported. And you can even do engaging things like make predictions of what it's going to be like in a particular year based on what people have said in the past. So the things that you definitely need if you are interested in starting a program, definitely consider it as a long-term effort. It often will take at least a year to sort out the kinks and like try out a site. Maybe it won't work that well the first year. Maybe you realize like, oh, we were way too ambitious. We tried to add 10 different species with like 3 plants each over this huge trail, and no one wants to do all the observations. So we need to scale back. And just like discovering that kind of thing will happen in the first year. So definitely think of it as a long-term commitment. You'll definitely want to also have a leader, someone who's on the ground. This doesn't have to be a professional or paid person at an organization. Having a volunteer who's in that role is great as well, but someone who's kind of dedicated to be the one who's going to help with the scheduling. If you're doing, a schedule to do rotating observations, if you're going to try to have someone that's doing some like training, we have a lot of training resources on our website, but having someone who's there who can like go out in person is really valuable. Because we're a very small national coordinating office of 6 people for the whole country, we have to make sure that we have all these resources on the website, and we really rely on these leaders to be our people that we do train the trainers with, and then they train others on the ground. And I think it's also helpful to have someone who's kind of the data person too. We do a lot of data sharing. So I mentioned all the campaign newsletters. You can leverage those, sign up for a few campaigns and then share back those with your volunteers, or even have your volunteers sign up for those newsletters. But someone who's going to at least be accountable for saying okay, let's take a look at what we have here, I think really helps with sustainability of your program, because it feels like, oh, we're working towards something. We have a question, we're trying to answer it, and this is where we are now in that process. Okay, so I mentioned the local phenology leader course. We're about to launch that for the spring. It's going to start on March 8th. So the applications are now open as of yesterday, I think. We actually haven't even sent out our email yet to the interest list. I think that's going out tomorrow. But it is $55. That gives you a hard copy of the botany primer if you don't already have one, as well as a name tag. That's a Nature's Notebook name tag, and then a certificate. So you can sign up for that if you want. We're also going to be offering a self-paced option this year, where you won't have the in-person time with a cohort, but you'll be able to work through the materials that we have in the course. So that's an option for this year. Okay, so a couple things about some of our key projects. So Desert Refuge is a project that we launched a couple years ago in collaboration with the Desert Botanical Garden. This is funded by Monarch Joint Venture and the U.S. Forest Service International Programs. And this effort involves what we were doing basically earlier with the photos. You find a milkweed plant and you spend 10 minutes a week looking at your plant and looking if there's any activity of monarchs or caterpillars on your plant. So it really only takes 10 minutes a week, and we're interested in recording this now through about May. It's unusual here, which is why we're doing this project, that monarchs are actually active here in the wintertime. They usually leave in the summer because it's too hot, but we do get monarchs, not just the migrating ones that are passing through, but we have resident monarchs that are actually breeding here in the wintertime. And we have this is possible because of the milkweeds we have here are actually alive, growing, and have leaves and flowers that attract the monarchs. So there's a lot of interesting questions associated with this campaign on the website there. But if you have a milkweed, this is a really great one to participate in. We have a couple invasive species efforts. These are not so much long-term observation things in Nature's Notebook. You can track them in Nature's Notebook, but because they're invasive grasses, we don't expect you to look at it week after week and not get rid of it. So we have a one-time reporting form for this one. And so if you see bufflegrass when you're out and about, go on this link on your phone. It'll auto-locate you if you say that that's okay, and then put the data in. So all you have to answer is, do you see green leaves on the plant and how much green is there? And there's photographs to help you. And then we have a similar thing right now for red brome, if you have areas with red brome. And then I think this is the last one. So another effort we just started last year called Project Lucy Tree is involves, if you have a mesquite tree near you, that's ideal. They're interested in understanding if there's something about the mesquite's phenology that's attracting the Lucies to come and nest in mesquite trees. If it has to do with the stage that it's in with the leaves or the flowers. So that's ideal. If you have a Lucy's warbler nest box already and you don't have a mesquite tree, that's okay. You could still do the surveys and then just write where your closest mesquite tree is to where your nest box is. And then you can report that in Nature's Notebook. And we have more details about that as well. So I think that's all I have. I know it was kind of a lot at the end there, but this QR code is for our newsletters. We have 3 of them. There's an observer newsletter for anyone who's a Nature's Notebook observer, a partner newsletter that's for local phenology programs and other partners, and then a researcher newsletter that's more geared towards researcher tools and opportunities and things like that. But I'm happy to take questions, and I put my email there too, if anyone wants to reach out later about local phenology programs or anything else.
There was one question that says, what if you live in one place for 6 months and another place for 6 months. Is it still acceptable to set up a backyard site?
Yeah, definitely. So I would try to pick something to observe that you can at least record like the start of it or the peak of it if you can, even if you're not going to be there for the whole thing. So like some of our campaigns are focused on like recording the like pollen trackers is focused on flowering. So if you know like I'm going to be up in, I don't know where you'd be, like in the Northeast, and I'm going to be there through April, then you could record maybe like the start of flowering. I guess you would be there later. You would be there like April or May, right? So I don't know if you would miss out on a phenophase, maybe then you would try to do something like record the fruiting of that tree, because you'd be able to capture the start of it. We're most interested in when things are starting and when they're ending, and then the peak. So if you can get like at least one of those pieces, that's probably the most useful. So you could be tracking two different things in the different places. And then maybe if you're here in Arizona during the winter, you could do like desert refuge, and track a milkweed, because that's probably going to happen in the period you're here.
And then one more, Sean was wondering if he could get the R package info.
Oh, sure. Yeah, let me find that and I'll put that in the chat. Yeah, and we if you have any questions about that package, feel free to reach out to me. I'm not an R person, but I can point you to people who are that can help you.
And I think that's all for questions, unless anybody else has any.
I would like to ask Aaron and perhaps Kathy about the project that was set up for Maricopa County Master Naturalists.
I mean, it's like a
Go ahead.
group. People just have
Yeah,
to
so
set projects up under it. It's like, it's kind of just sitting there. I just updated the email to go to our Gmail account, but
cool.
yeah, you can probably explain it better, Aaron.
Yeah, well, I'm not sure about the history too. I could look back at my original
Well,
email from you guys, but yeah, group is kind of synonymous with local phenology program. We've been moving more towards local phenology program, but it's basically, yeah, it's the functionality of having the ability to have multiple people observe at the same site or sites. You can have multiple. And once you have a local phenology program, you get this is a profile page. This is the thing on our new website. If you want to join this, you would have to be a Nature's Notebook observer, but you join the program with this button. If you don't have an account, it'll say log in or register, and then you can do that first and then come back. But once you have a program, you get all these tools here. And so it'll give you the contact information. There's a bunch of tools here. You can visualize the group data. We say group here still. Visualize the data in the visualization tool filtered for this local phenology program. You can download just the data. You can look, this is more like participation metrics. You can see the members that are part of it and manage them. There's a whole bunch of resources, including the certification course. These two tabs are only visible to administrators, just so you know. So you have to be logged in as an administrator of the group like Kathy is to see them. But we have lots of resources here, program planning stuff. Oh yeah, the program planning stuff's on this tab. So lots of stuff available on the profile page.
We originally used it when I had run one of the courses. I had set it up. I think it was the one like Kevin, and I don't remember who else was in that one, but I had them go out and like find a site and like practice using it. And so they kind of, got practice that way. So, and initially my idea, it never happened, was for us to have phenology trails at all the Maricopa County parks. And then, the master naturalists could monitor them. And then maybe we could eventually get the public involved. It never happened because I didn't have help. So I had too much going on, and it kind of, nothing happened. But yeah, but it is there, and I'm happy to like give anybody admin access if, Karen, if you need it or whatever. Like, I don't know how I'm assuming I can go in there and do that.
Well, I know that 2 years ago, Andrea Michaels and Beth Slikis Andrea, I think you are here this evening. Do you want to, you had plans to set up a phenology trail. Was it at Cave Creek?
It was at Cave Creek Regional Park. We had enormous plans as part of our midterm, and had absolutely no concept about the depth of planning that it would would take. But we were extremely enthusiastic, and wanted to involve community college, as volunteers, and it was all working with Juanita as a complement to their study after the wildfire.
Mhm.
And we were going to get families and everybody involved.
So did it feel like the program planning process was a little daunting?
The program planning was daunting. We had lots of stuff to do, and the park is about 45 minutes to an hour away from us. So that was a part of the problem. And then we had this big idea about in May, and I live in Flagstaff for the summer and then came back late. So
Yeah,
It's-
definitely the location of your site is really important. And it has to be convenient enough that you'll be able to go there. I think that's one of the main things that causes programs not to work out. It's just like it's too ambitious, it's like too far away. You're trying to do too much with too many plants or animals, or the area you have to cover is just too big. So yeah, I think trying to start as small as you can is best. And I think, like Kathy was saying, having people practice and get familiar with the platform in your own backyard is a great way to start.
Yeah, it looks like I set up, I don't remember, sorry. I've got some memory issues now, but Cave Creek Regional, Santan, and Veterans Oasis were 3 sites that I set up. I don't remember why I set them up, but they have plants in them. So I clearly did it for some reason, but anyway, I was just looking to see what was in there.
Yeah,
So
well, we can
Kathy, now
always-
that we have park stewards from our master naturalist previous cohorts, I'm calling them park stewards. I think that's what Juanita refers to them. But at most of the parks, not at Santan yet, but at White Tanks, Estrella, Usury, and I think there's another one, we have master naturalists who are kind of overseeing a lot of those volunteer, like desert defenders and such. So this might be a good time to reactivate that with some interest, because we have more assistance now to help.
And Santan has a rush milkweed. I know that because I was monitoring it. It was one little lonely one.
Nice.
Just all by itself.
Yeah, and
So-
if you already have a program that's set up, but you want to like adjust it, we can work with you to do that. Like if you want to, like, I want to keep this plant because I still know where it is and I want to keep the data set going, we can move it to another site for you, or we can like we can rename sites, we can move stuff however you want.
I thought that the idea of having a small phenology trail near the visitor center would be so nice
Mhm.
at Cave Creek Park, because there's a little bit of water there and it's a nice, easy trail and quite a lovely variety of plants. And if there were like a simple phenology trail that any family with kids of sort of any age to just introduce them to the idea of phenology, maybe it would keep them, if not coming back to the park on a weekly basis, at least maybe on a monthly or quarterly basis, that they would find it fascinating to see how the things they were observing changed.
Yeah, I don't know if it's still going on, but Hacienda did an amazing job with theirs. They had like a display board with information in a book that you could like check out and go do the monitoring. And they very heavily were like engaging the public on it. I'm not sure if it's still going strong and stuff, but they had the trees marked very clearly with tags. it was wonderful. That's probably the best I've seen as far as like engaging the public. And I don't know, like South Mountain had a good one. I don't know, Aaron, can you take over a phonology? Like, let's say it was like South Mountain used to have one who used to be our old sponsor a long time ago. And it was really, really like they had a lot of data. But then, of course, the center kind of closed. Can you revamp? Like, is there a way for, like, if we wanted to reestablish that, can you, like, can someone take that data and reestablish it?
Yeah, I think usually the limiting thing is like knowing where the plants are that are all on the list.
Okay.
But
That's, yeah, that's
if
true.
you can figure it out, like sometimes the tags are still there, or if we can get a hold of the person that was involved, or
Yeah.
sometimes there's like a map out there. Yeah, we don't have like GPS points for each individual plant, but if you kind of know like there's only one milkweed, so it's got to be that one, you can keep it going. But that's usually the thing that limits doing that kind of thing. But I mean, technically, we can give administrator access to a new person.
Okay.
Okay, I don't think there were any other questions, so let's give Erin a tremendous thank you for incredible information. We've had a lot of questions about phenology from this cohort, and I'm seeing potential for capstone projects, which we'll talk about briefly before we break into our mentor groups, because I think there's some real potential here on the part of some of ours to help bolster Kathy's project and also bolster what's happening at NPN. So thank you, Erin, so much for sharing your evening with us. Valuable information, and we'll get you some data.
Awesome. Thank you. Yeah, thanks for the great engagement and questions. And yeah, I really feel free to reach out if you all want to talk more about the local phenology programs, any questions about that.
Thank you.
Right.
So what you just heard, kind of this discussion, is a potential for the capstone project. We've been getting lots of questions from people about what should I expect. So we will delve into this a little bit more each week, giving you ideas, but phenology trails are a great way to get kids and adults. Frankly, how many adults are in tune with nature these days? We live in such an urbanized world. The idea of using plants that are already there around visitor centers at our parks, especially those parks where we already have some veteran master naturalists who are serving as kind of a quasi park steward, helping organize events. Lots of different possibilities here. So if phenology is something that piques your interest, and this doesn't have to be a solo project. As Andrea shared, she and Beth worked together on that project, and they put in a lot of hours in it, I think not realizing what they were getting into, as she said, when they first started. But now that a few things are established, and just slicing off a small area around a visitor center, mapping what's there, and getting, working with, I guess they would work with Juanita if they wanted to put up just some like temporary signage of some kind, just to indicate a plant under study. It seems like there is is it Cave Creek that has some markings? Somebody who's been there recently has markings around the visitor center on plants. One of our parks had markings for some plants that were under study for something. Nobody knows? Okay. Well, anyway, that is one possibility. So, and while he's here, Dave, why don't you share what you did for your capstone last year?
Yeah, so I made a box for tabling events that sort of just has a bunch of things inside of it that you would use as a master naturalist. So like field guides, first aid, geology maps, magnifying glass, basically just a little bit of everything from all the different classes that we had, and I put it all together in one box. Yeah, so that you can use it to talk about being a master naturalist at public events.
So if you're doing tabling events, that box resides with Sue. I don't know which event it will be at on February 24th, but we only have one box, but it was great. And we saw the Arizona flag that Dave painted onto it. So you'll have one of our state symbols, or at least our state flag, there as well. Okay, with that, I think it's time for us to break into our mentor groups. I don't believe Sharon was able to be with us tonight, so I think that was room 3.